Okay, good day, everyone. My name is Elliot Raises from the CDC, and I am as a member of the organizing committee for the 30th International Workshop on HIV Drug Resistance and Treatment Strategies. I want to welcome you to this pre-workshop webinar that we are having today. As you can see, we plan to have a in-person meeting in Cape Town, in beautiful Cape Town, South Africa, April 9th through 11th of next year. But what the organizing committee has decided to do is, as a build-up to the meeting next year, 11 months from now, we want to have a series. We're going to provide a series of webinars, with today being the first of a series of webinars with an exciting program that you will hear more from other panelists in a few minutes. So keep in mind, please save the date. We would love to see you in Cape Town, South Africa. It really is a wonderful meeting, and much of the information that you're going to see shared today was first presented at that meeting. In addition, so just to give you a couple of dates, registration for the meeting will open in September. And also, we would like you to start thinking about any research you may be doing related to HIV drug resistance and or treatment strategies that you might want to submit an abstract. Abstracts will start being accepted in August of this year. We have a long abstract acceptance period all the way until January of next year. So, but we think that it's an abstract-driven meeting with a lot of good plenaries, as you'll see for more information on the website, and follow us on LinkedIn and all sorts of various social media haunts. So with that in mind, I'm going to turn the session over to my co-chair for the webinar, Eva Avalos.
Thank you so much, Elliot, and thank you all again for being with us. I have the pleasure of introducing our moderator this evening, who is the legendary Dan Kuritzkes. I think most of you know him, but for those of you who don't, Dan received his BS and MS degree in molecular biophysics and biochemistry from Yale University and his MD from Harvard Medical School. He completed his clinical and research training in internal medicine and infectious disease at Massachusetts General Hospital and was a visiting scientist at the Whitehead Institute for Biomedical Research before joining the faculty at the University of Colorado Health Services Center. Dan returned to Harvard Medical School in 2002, where he is now the Harriet Ryan LB Professor of Medicine and Chief Division of Infectious Disease at the Brigham and Women's Hospital. Dan has published extensively on antiretroviral therapy and drug resistance and HIV infection. He has chaired several multi-center studies of HIV therapy and previously chaired
Looks like Ava froze here.
You may have lost Ava, so we'll go ahead and turn it over.
We'll just go ahead. Thank you very much for that kind introduction. And I think we'll just move on with the program. So it's really a pleasure to be serving as the moderator today. We have really an outstanding panel of international experts in HIV drug resistance going to be speaking. And it's my pleasure to start off by introducing Professor Gert van Zyl from Stellenbosch University. Professor van Zyl is a virologic pathologist and professor in medical virology at Stellenbosch University and the executive head of the Department of Pathology at Stellenbosch, as well as the National Health Service Laboratory in Tygerberg, South Africa. He studied medicine at the University of Pretoria and specialized in virological pathology at Stellenbosch, obtaining his PhD by investigating HIV drug resistance also at Stellenbosch. His research areas focused on HIV drug resistance and HIV persistence, doing a sabbatical visit in John Meller's lab at the University of Pittsburgh about a decade ago now, and also collaborating with Mark Cotton, who was the co-lead of the pediatric SHARE trial. Other research areas are third-generation sequencing and viral diagnostic test methods, and much of his research relates to diagnostic service delivery in resource-limited settings, which often require novel solutions. working as he does at the interface between clinical care and laboratory diagnostics, Professor Van Ziel finds himself in the privileged position to work with excellent adult and pediatric infectious diseases clinicians and international and South African clinical and translational scientists, and most enjoys working in teams that include young postgraduate students and has successfully supervised 6 doctoral students and several master's students. So we'll hear today about dolutegravir resistance in the TLD era.
Thank you very much to the organizers, and thank you to Dan for the kind introduction. It's a big privilege to share this, and this emanated from a theme discussion at CROI, which I was very privileged to chair, and where the real champions here are the abstract authors who presented their data at this session. We've seen that dolutegravir has been recommended by the WHO for both therapy-naive and therapy-experienced patients. And this is largely due to the high success rate in treatment, both for first and second line, and also the high genetic barrier that's observed when this, especially when this regimen is used in treatment-naive patients, with very few patients developing drug resistance. But there were recent reports of increased levels of drug resistance, and this emanated from an unprecedented rollout of dolutegravir regimens in resource-limited settings. And patients are often transitioned to these regimens without viral load monitoring, and viral load monitoring is often done infrequently in these settings. And people are spending increased time on dolutegravir, which was introduced as early as 2017 in some Southern African and some countries in Sub-Saharan Africa. And this all could have contributed to these emerging reports of drug resistance. We know that drug resistance emergence is a result of a complex interaction between the virus, the treatment regimen, and the host, of which many things are important. Probably we have people like Catherine Orrell here, adherence being one of the most important. Drug resistance data is often difficult to collate, and it's also important to notice what is used as denominators. When people report drug resistance, they could use everybody treated with a dolutegravir regimen, those with virologic failure, those who have drug resistance testing, and then the numerators can also differ. People can report any drug resistance mutations or major mutations using either the Stanford drug resistance list or the IAS drug resistance mutation list, or the level of resistance often reported as intermediate or higher by investigators. The IAS drug resistance list report particular mutations as associated with a higher risk of drug resistance to dolutegravir. And the major mutations are listed next to dolutegravir. Of these, a recent review by Bob Schaefer's group showed that in particular 3 mutations are associated with commonly occur when people started dolutegravir as their first integrase inhibitor. The Stanford drug resistance list largely corresponds with the IAS mutation list, but 2 mutations are provided in addition and at position 66 and 92, and 1 mutation is omitted at position 153. Studies of actually drug resistance are also different, and each kind of study has its own benefits. Randomized controlled trials are often regarded as the gold standard because allocation is unbiased, but there are strict entry criteria, so patients with advanced disease are often excluded. And because of the cost, the follow-up is often short, and drug resistance evolution can take long. Population-based studies, observational studies, have the benefit that people can be observed over a longer period, but there's no random regimen allocation, and the samples can be convenient samples and are often not representative of the population. Laboratory-based studies are the least costly and often pragmatic, but the population denominator is unknown, and there are referral biases. But they can be valuable in observing longitudinal trends. So if one uses the same methodology over time, you can at least see whether there's an increase in drug resistance. There are also different treatment scenarios when people use dolutegravir in a treatment combination, and I'm just focusing here on triple combination therapy. Treatment-naive patients have the highest treatment success and an almost negligible risk of resistance, especially if their baseline viral loads are below 100,000 copies. Then patients who transition from first line, when their viral loads suppressed, they also have a very low risk of resistance and a high treatment success rate. But patients who are viremic are at increased risk, and they can be similar if they had long periods of viremia to people who have experienced treatment failure, which is the next category. So treatment-experienced patients, often in our context, people who switched from an INSTI regimen, still have high treatment success rates, but the risk of dolutegravir-resistance has increased or are increasing in these groups. And then the highest risk is in integrase inhibitor experienced patients, the highest risk of failure and the highest risk of drug resistance. So the abstracts presented at CROI in this session, the first one was presented by Richard Lessels and covered groups from Zambia and Malawi in 2 cross-sectional studies. And this was the abstract title. And they studied, they had 2 main outcomes. The one is to assess the risk of viremia above a viral load of 400 copies per ml, and drug resistance up to 2 years after programmatic switch from NNRTI-based regimens to dolutegravir. The setting was in Malawi Lighthouse and Zambia Center for Infectious Diseases, and the participants were adults enrolled at the time of routine switch from NNRTI-based therapy to DTG ART regimens. And they were followed up at 1 year and 2 years. Almost 1,500 participants were enrolled in both groups. One should note that very few patients were viremic at the time of switch, both in the Zambia and in the Malawi cohorts. And few patients, and the majority of patients had a valid viral load measurement at the 2-year follow-up. Only 3.2% of the patient overall had viremia at the 2-year study endpoint. And looking at the figure on the left, one could see that men had a significant higher risk of virologic failure, and patients who had a high baseline viral load or were viremic at the time of switch had a much higher risk of odds ratio of having virologic failure at the 2-year time point. And then also Zambia showing a higher risk of failure. Only 2 cases had drug resistance, but one should note that this is really representing what we now call a TLD1, largely the use of TLD in treatment-suppressed patients, and very few patients were viremic. So to conclude, most patients remained virologic suppression at 2 years post-switch, a low rate of viremia overall, good treatment success rate, and few cases with dolutegravir resistance. The next study was from Kenya, presented by Dr. Kingwara, Leonard Kingwara. And they investigated patients, or the context was that Kenya started dolutegravir rollout already in 2017, and they used remnant samples from the Cadre surveillance study. patients were eligible when they had a single viral load of above 200 copies per ml. There were two main groups. In green are therapy naive, and in this beige orange color, treatment experienced patients. The treatment naive were patients who were initiated on a dolutegravir regimen, and the experienced ones were transitioned. They did not provide data on how many of these patients were viremic at the time of transitioning. But he reported during the conference that viral load monitoring cannot be done too frequently. Of the treatment experienced patients, 7 out of 31 had INSTI mutations, whereas only 1 of the 12 naive patients had INSTI mutations, showing the higher risk in patients who are treatment experienced. The mutation list are shown here. What is notable is the relative high frequency of the E138 mutation, which is not one of the mutations that was highlighted in the review by Bob Schaefer. So there might be differences related to different countries. So to highlight out of his presentation was the high frequency of drug resistance in INSTI experienced patients. And he concluded that this emphasizes the importance of close monitoring with viral load and drug resistance testing in patients who are failing on dolutegravir, not as when they start this, when they're already treatment experienced. The next study was from Lesotho, presented by Dr. Labart, and titled Emerging Drug Resistance in Lesotho. They investigated this in doctor-led and nurse-led sites, and the criteria for drug resistance testing were either 2 viral loads above 50 copies per ml or 1 viral load above 500 copies per ml. And these patients were transitioned from NNRTIs to DTG. They included about 15,000 patients, of which about 1% had qualified for viral load, for drug resistance testing because of an elevated viral load. About half of them had successful sequencing, and 8 out of 78, about 10%, had a DTG drug resistance mutations. There are 3 figures shown here. The top figure indicates the viral loads. One could see that 6 of the 8 patients had an elevated viral load before they were switched. So 6 of the 8 with drug resistance. The second slide shows the longitudinal decrease in dolutegravir susceptibility over time as more mutations accumulated. And the last figure, the bottom one, shows that there was not significant changes in the NRTI susceptibility over time. The next slide, or the next presentation was from Mexico, so the only one not from Africa. And this was a laboratory-based study. They presented data from a population of 87,000 patients on second-generation INSTIs, Bictegravir and Dolutegravir regimens. Of these 100, 75 were from treatment-experienced patients who were on a second line, and 25 were from patients who received integrase inhibitors as a first-line regimen. And of these 25, 5 had primary drug resistance mutations, and in the treatment-experienced patients, 15 of 75 had primary integrase mutations. The most common mutations were 263K and N155H in the treatment-naive, and combined with positions 140 and 148 in the treatment-experienced patients. He highlighted that these samples were tested over a 27-month period, that only 20% of them had primary INSTI mutations. The most common mutations were highlighted. And an interesting observation was that the majority of the patients with a combination of 263K and M51I had relative low viral loads, and that none of the patients with primary resistance had M194V, which was, I thought, a surprising finding, probably indicative of inadequate adherence in many of these participants. The last presentation was from South Africa, from Cape Town, presented by Ying Zhao, and this was a prospective cohort study. Patients who were treatment experienced and were on a PI regimen with virologic failure had genotypic drug resistance testing when they were on these regimens for more than 2 years, according to the South African guideline. And they investigated and they compared the outcome if patients were treated with TLD versus a second-line PI regimen. The options when they were failing on the PI regimen depended on whether there was PI drug resistance. If there was no PI drug resistance, they could remain on their regimen with adherence support, or they could be switched to TLD. But when they had a PI resistance, they had to switch to darunavir, or could also be switched to TLD if they did not have increased if they were fully susceptible to darunavir. So the outcomes were to look at virologic suppression. That was the primary outcome. And the secondary outcome was to look at the emergence of integrase drug resistance. So the population of the 355 consecutive cases, 133 were switched to TLD, 121 remained on the second-line PI regimen, and 101 were switched to darunavir. In the majority of these cases, the patients also received dolutegravir with darunavir. Of the 133 switched to TLD, only 4 patients had TDF resistance. The majority had no TDF resistance because that was the initial guideline decision. One could clearly see, if one looks at the proportion with probability of suppression over time, that there was no significant difference between TLD and darunavir in the groups which were switched, and then TLD, and in those without PI resistance, comparing TLD with remaining on a PI regimen, a much better outcome for those who switched away from the PI regimen. And this could largely be ascribed to the fact that patients were able to adhere much better to TLD than to their PI regimen. The conclusions were that those failing a second-line PI regimen without PI resistance benefited from switching to TLD because they had improved virologic outcomes. Virologic outcomes were similar in patients with PI resistance switched to darunavir compared to those who were switched to TLD. And there was a low incidence of treatment emergent resistance. Of the treated population, only 2% developed drug resistance of those treated with TLD at 1 to 3 years after switching. And this provides support for TLD as a third-line regimen or alternative second-line regimen after PI failure. So to combine the findings from these studies presented at CROI was that there were big differences between patients who were effectively receiving TLD1 as a first line, or those who received it in treatment when they were already treatment experienced, either viremic switches or after failure on an NNRTI regimen. In Malawi, most patients effectively received TLD1 because very few were viremic when they were switched. and there were only 2 cases of INSTI drug resistance. In Kenya, we don't know how many of them had virologic failure, but a much higher rate of drug resistance were observed in those who were treatment experienced. In Lesotho, only 10% had drug resistance, and 6 of these were viremic on an in-and-out regimen before switch. In the study from Cape Town, there was only a 2% population risk of TLD resistance. And then to contextualize this, in a recent review by Chu et al. With Bob Schaefer as senior author, this showed that in patients switching from 2 NRTIs to NNRTI to a DGG-based regimen, that only 1.5% have drug resistance if one look at the whole treated population. And of those with virologic failure and drug resistance testing, 20.6% have drug resistance with INSTI mutations. What I think we can learn from this is that the factors that are increasing the risk of DTG resistance are INSTI experience versus naive. Now, this emanates from other studies, but also treatment experience has a higher risk than treatment naive. If they are viremic when transitioned to DTG, their risk is much higher than when they are virologic suppressed. The longer follow-up, we could see an idea that there were more cases with virologic failure at the 96 weeks compared to the 48 weeks. Regimens, monotherapy, from other studies, we know monotherapy has a much higher risk of resistance than triple therapy. And in patients with viral load, many studies have shown high baseline viral load increases your risk of virologic failure and hence also drug resistance. And then ongoing virologic failure, if they do not improve with adherence intensification, if failure persists, then they are at higher risk for drug resistance. And we have learned, and we're starting to learn that also in programs, that we should not wait for patients who have continued failure. And then advanced disease, other studies, and especially observational studies had shown that low CD4 count is associated with an increased risk of resistance. And one wonders whether HIV subtype might influence this, as was observed for Cabotegravir injectable treatment. I want to conclude by saying that that the main benefit of dolutegravir regimens is probably not so much its high genetic barrier, but the high virologic success rate. Because there's very low resistant risk when people start with a triple DGG regimen. Population level resistance rates in patients on TLD2 or equivalent remains low, but if patients experience ongoing viremia on DTG regimens, there is a high risk of resistance in these individuals. TLD2 or equivalent patients require active viral load monitoring and resistance testing when viremia does not resuppress, and we cannot ignore viremia in these patients. And INSTI mutation prevalence in treatment-experienced patients varied geographically, and this is probably related to virologic monitoring and the indications for HIV drug resistance testing, and possibly genotypic differences. I want to acknowledge the authors who shared the slides for the theme discussion, the CROI conference organizers, and advice and input from Bob Schaefer, who authored recent reviews on the topic. And thank you very much, and thank you for the opportunity to share this.
Thanks so much, Gert, for that really terrific overview. I think that sets the stage for the rest of the session today very, very nicely. Before I introduce the next speaker, let me just remind all of our audience to please submit your questions through the Q&A link that you see at the bottom of your screen. We'll be having a panel discussion after hearing from all of our speakers, and really look forward to being able to answer your questions and have our panel members discuss them. So it's now my great pleasure to introduce Professor Nick Paton. Professor Paton trained in medicine and infectious diseases really around the world, in Cambridge, at Sydney, and London, as well as in epidemiology at the London School of Hygiene and Tropical Medicine. For many years, he was the head of the department of the National HIV Referral Center in Singapore, and there developed the Center for Research in HIV and Communicable Diseases. From 2006 to 2011, he worked at the UK MRC Clinical Trials Unit, where he was the chief investigator of several large HIV treatment trials, including Pivot and Earnest, which remains one of the key studies that we refer to with respect to the impact or lack of impact of resistance in second-line therapy. He currently holds a joint appointment as Professor of Infectious Diseases at the National University of Singapore and at the London School. He's the chief investigator of the Nadia trial, another one of those essential studies, as well as the scientific lead of the CARES trial, about which he'll be speaking shortly. In Singapore, he leads a program of TB trials focused on studies of host-directed therapies for TB and exploring novel TB trial outcome measures, and is the chief investigator on the Truncate TB trial, a strategy trial of 2 months of treatment for drug-susceptible TB done across a network of sites in Asia and Africa. We'll be hearing today about Cabotegravir, rilpivirine, and other long-acting agents in resource-limited settings. Nick?
Great. Thanks very much, Dan, and thanks very much for the invitation to speak at this meeting. Now, I'm trying to share. I'm assuming this is full screen presentation mode, but please shout if
Looks great.
Okay, great. Thanks very much. So here are my disclosures. So this is what I'm going to cover in the next, I guess I've got about 15, 20 minutes. Mainly Cabotegravir, rilpivirine long-acting, talking about the evidence gaps before we can roll this out into resource-limited settings in the public health approach. Talk about the main findings from the CARES trial, and also explore a bit more about subtype and resistance issues. And then talk about the remaining data needs and barriers to implementation, particularly around hepatitis B, non-adherent populations, and other, I should say, NNRTI failure. And then just wrap up with, since I was given a title that was broader than Cabotegravir, rilpivirine, just comment on some of the other things that may be moving forward, or in either in the near future or in the distant future, in resource-limited settings. So first of all, before the CARES trial, and really what informed the CARES trial, was that there were a number of evidence gaps for use of long-acting therapy in resource-limited settings. Really, we had to ask the question, is there sufficient trial data in appropriate patient populations? Is there sufficient trial data with appropriate monitoring, with appropriate healthcare delivery systems? And then do we have enough to determine preferences, quality of life, and cost effectiveness? All of this really thinking about, could this become a recommendation in the WHO treatment guidelines for resource-limited settings using the public health approach? And if we look, and this is a nice review that's just come out by a colleague, Ivan Mambule, who's based in Uganda, in Lancet HIV, where they reviewed 2 drug regimens, but one of the tables summarizes nicely the Cabotegravir, rilpivirine data and trials up until CARES. And if we look at the recruitment in Africa, LATE, nil. In Atlas, there were 7 to 9 out of 100+ sites. In FLAIR, there was 8 out of 100+ sites in South Africa. All in South Africa, no low income countries, no low middle income countries represented. In terms of the proportion of Black participants enrolled, that was also certainly low compared to the obviously the population in sub-Saharan Africa. So in addition to representation of race, gender, sex is critical. So generally the trials have had underrepresentation of women. People in sub-Saharan Africa likely to have occult resistance to non-nucleosides from prior episodes of undetected treatment failure, or nevirapine for prevention of mother-to-child transmission, different viral subtypes, different comorbidities, concomitant medications. And critically, none of the previous trials had actually compared cabotegravir-rilpivirine with TLD, which is the current almost universal standard of care in resource-limited settings, particularly sub-Saharan Africa. What about the monitoring approach? This is the Atlas trial schedule, but it's typical of all of the trials preceding CARES, with essentially monthly viral load monitoring for the duration of the trial and similar frequent safety monitoring, which is at odds with the WHO's recommended 6 to 12-month viral load monitoring. And it may not be obvious, but what's the problem with that? Well, first of all, the trial approach may not be feasible to implement in the public health approach if you're using very frequent viral load monitoring. It may be too expensive, but also a trial doesn't quantify the potential risks in the public health approach. If you're doing excessively frequent viral load monitoring, you're not going to capture the risks of people who are only tested every 6 to 12 months, who may have prolonged periods of viral replication with ongoing treatment and be at higher risk of resistance. And similarly, you're not going to potentially capture important safety events if you're not allowing periods of allowing frequency of monitoring that would be typical in the public health approach. So it's important, really important, to do the monitoring in a way that's relevant to the rollout. Appropriate healthcare delivery systems, Atlas, Flare, all of the studies look fantastic in terms of getting people to have their injections as scheduled. Very few people outside of the 7-day window, a handful above 14 days delay. And then, of course, differences in healthcare delivery system go beyond just coming back for the injections. The resources to schedule, to remind people to come, and the patient perceptions that may drive adherence or non-adherence may be different. Cold chain, facilities for administration of injections, and so on, so many differences. And then the perceptions of participants who require treatment for HIV and factors that impact quality of life. All of these things are different. So that was really the drive for the CARES trials, trying to generate the evidence that we need for potentially this to move into a WHO guideline recommendation to use cabotegravir-rilpivirine in the public health approach, or at least a first step towards that. And let me just start off, because at the end of the presentation I'll go into a bit more discussion, but let me start off by acknowledging the CARES trial team in Kenya, Uganda, and South Africa, a very large team. I've listed the main investigators here. It was funded by Janssen with support from ViiV, and of course acknowledging the trial participants who really did a great job in sticking with the protocol and giving us a very robust data set. And importantly, acknowledging Sisi Kichio, who was the chief investigator, and Ivan Mambule, who's the project leader. And the trial was run coordinated out of the JCRC in Kampala. So here's the CARES trial design. It's phase 3B, randomized, open label, active control, multi-center parallel group, non-inferiority trial. We look first of all at the eligibility criteria. People had to be on stable oral therapy with a tenofovir-based regimen, with the third drug being dolutegravir, nevirapine, or efavirenz. They had to be undetectable viral load at screening and have an additional viral load 4 to 12 months earlier, which was also undetectable. No history of treatment failure. In many cases, that's difficult to ascertain accurately, but that was the expectation. And no hepatitis B infection. And as I'll talk about later, that was done by testing for surface antigen and for anti-hepatitis B core antibody. So both of those tests needed to show absence of hepatitis B exposure or active infection for the participant to be enrolled. They were randomized to continue standard of care or to cabotegravir-rilpivirine with 2 monthly injections with an optional oral lead-in period of 4 weeks. Very standard. This part is very standard for all of the trials. But what differs with CARES, as I've given you in the preamble, was the viral load monitoring was every 24 weeks. We also, and safety monitoring, similar sparse safety monitoring, we stored PBMCs at baseline to look at proviral DNA and sequence that for subtype determination, archive resistance. And the primary endpoint is at week 48. That's what was presented at CROI, and that's what I'll just run through briefly. Now, it's going to be actually published in a couple of weeks' time, I'm pleased to say, in Lancet Infectious Diseases. So I'm only going to skim through it today, but there's lots more detail in the paper and in the supplementary appendix about the things I'm going to talk about. So primary endpoint, week 48, the trial is ongoing, nearly finished, week 96 follow-up, and that data is going to be important too. So the primary outcome of CARES was the plasma viral load less than 50 copies per mil at week 48, assessed using the FDA snapshot. It was non-inferiority. We set a 10% non-inferiority margin, and then we did a sensitivity analysis in the per protocol population, but the primary assessment non-inferiority was in the ITT. And also looked at viral load above 50 copies, confirmed virological failure, and key treatment satisfaction, and obviously safety. So the trial was enrolled in Uganda, 3 sites, Kenya, 3 sites, and 2 sites in South Africa. We randomized 512 participants, very few withdrawals. So 4 people out of those 512 withdrew. So very high retention at week 48. So it's a robust data set for drawing conclusions. Here is the adherence to injection, and it looks pretty similar to all of the other trials. So within these clinical research sites in sub-Saharan Africa, there was really very good adherence to the injections, and just 9 people had an injection delayed more than 14 days, and that only applied. It was only 1 injection per participant. Nobody had serial delays in injections. So the baseline characteristics, majority female, 58%. You can see 21% had a BMI above 30, so obese. Essentially everyone a black race. Prior exposure to non-nucleosides in 74%, although virtually everybody was on essentially TLD at screening. So many people had switched. And remember, these are people without failure, so they switched programmatically to TLD from an efavirenz-based regimen by and large. So here's the primary outcome. Essentially, the viral load suppression was high in both groups, 96, 97%. The difference between them, if we just look at the non-inferiority comparison, this is the CROI presentation, the final data set, that's about 0.8%. But well clear of our 10% non-inferiority margin. So we can declare that cabotegravir-rilpivirine is non-inferior to oral standard care, PLD, in this setting. And that was borne out in the per protocol analysis and also the analysis above 50 copies. So only 2 participants had virological failure. One of those met our criteria for being confirmed, which is 2 measures above 200. 1 participant, unfortunately, died before that second viral load could be done to confirm the repeat. But from the viral load was 40,000+, and resistance profile, it's clear that they would not have resuppressed if we'd repeated the viral load test. So essentially 2 people with confirmed virological failure. I've put their profiles here side by side, but on the left, the participant with confirmed virological failure at week 48, no delayed injections from Uganda, subtype A1, and here's the resistance profile at baseline. There was a 74M polymorphism, no rilpivirine resistance mutations, and at failure, the mutation profile there is shown a rilpivirine intermediate resistance, and then cabotegravir intermediate resistance, dolutegravir resistance low from that profile there. And this participant was switched to TLD and resuppressed on that, which I think is an important finding and key for long-acting therapy. We got something. If it doesn't work, we got something to go to, which is either retreat, or we've got from Nadia, obviously, darunavir TLD would work well too. The participant with unconfirmed virological failure had subtype D. Again, BMI was pretty just, was fine. At baseline, low level rilpivirine resistance, no INSTI mutations, and at failure, there was high-level rilpivirine resistance, a whole slew of mutations there, and they'd acquired a G118, which confers high-level resistance to cabotegravir and to dolutegravir. That's just been updated, I think, at Stanford. So clearly that patient would not have resuppressed. In terms of safety, there was pretty standard from all the other trials, so that there was an excess of grade 3 adverse events, and this is potentially interesting. The main difference, it was scattered, but the main difference was higher numbers of raised lipids, cholesterol, in the long-acting arm, which is probably the removal of the tenofovir. But in terms of events related to study drug, very few. So yes, there were more at grade 3, grade 4, but ones considered related to study drug, very few. And when we look at injection site reactions, typically as seen in other trials, all grade 1, grade 2, just one led to treatment discontinuation. So a reasonable safety profile. And the key thing is on the treatment satisfaction, what's the benefit for participants? It was clear that there was a strong preference, and certainly stronger outcome for treatment satisfaction in the people who are on the long-acting therapy, and we could potentially discuss what's driving that. So I've gone through the data, so I won't repeat this, but the overall conclusion, and I think this is important, is that this is the first demonstration of safety and efficacy of cabotegravir-rilpivirine long-acting. It's the essential first step to enable us to discuss, contemplate a potential role for long-acting in treatment programs in sub-Saharan Africa using the public health approach. It's not saying we've got everything we need now to move forward with this, but it was the key thing to at least show efficacy, safety, and some benefits before we could move forward. Now I'm going to just talk about some additional virology, just to elaborate a bit, because I think this is of interest, I guess, to this meeting and this audience. And I'm not a virologist, so I always get a bit nervous, especially when we have such distinguished people on the panel. Far more about this than me. But let's just look at predictors of failure, because I think this is a key contribution of the CARES trial. From the multivariable model, Chloe Hawken published several of these, but the most definitive one was this. 3 factors came out predicting virological failure on long-acting cabotegravir-rilpivirine. The first one was subtype A6A1, rilpivirine resistance mutations at baseline, and then obesity contributed to the mix too. Now, the concern with doing cabotegravir-rilpivirine in sub-Saharan Africa is actually subtype A1 is very common in East Africa. This shows, so red is actually subtype A, majority is A1. You can see Uganda, Tanzania, Kenya around here, but majority is A1. Subtype C down here in South Africa, Zimbabwe. So here are the viral subtypes in CARES. So actually, out of the 414, we managed to get an RT or an INSTI sequence from the PBMC as a baseline. 54% had subtype A1. There was no A6. And importantly, we used the Los Alamos reference lab as the reference strain. That's been an issue in the past with classification. And about 20% had subtype C and subtype D. So if nothing else, CARES is contributing to a much broader assessment of subtypes, but specifically this issue with subtype A1. And if we look at there's a strong predictor, A6A1, in this multivariable model of virological failure. So how do we reconcile that with the fact that we had such good outcomes in CARES? And I think the answer is here. It's shown in the supplementary appendix of this model, and it's discussed a little bit. Is that most of the driver of this A6A1 is actually A6 virus essentially from Russia, which was initially misclassified as A1. And the author's paper, that's the position on this. But when you look at the supplementary appendix where this data is, and it's certainly in the small print, so I've expanded it here. So out of the 199 people with A6A1, virtually all of them had A6. There were only 19 participants who had subtype A1 in this multivariable model in all of those combined earlier trials. 11, most of the virological failures in the A6A1 combined were subtype A6, but when you look in the small print, there were actually 2 with subtype A1 who met the confirmed virological failure criterion, and that actually is a higher percentage of the denominator than with A6. So I think it was a legitimate concern that A1 might be a problem. And so, you know, the data is actually somewhat reassuring that this model, probably this risk does not extend from A6 to A1. It really is A6. In a way, it's confirming the model. The other thing that's interesting, and there's no bias here in terms of picking a paper, there's a lot of, you know, I would say it seems to be a dispute in the literature and amongst virologists about the importance of L74I. His paper from Dan's group showing that actually there are biological reasons why this might explain a higher rate of detectable confirmed virological failure. And of interest, although the reviewers didn't let us put this in the paper in terms of highlighting this, because it is only one case, but we did not have any L74I in our participant population, but there was one case of L74M at baseline, and that was the one participant with confirmed virological failure. So it's N equals 1, but in the circumstances, I think that's very interesting. I mean, it's good to get people's thoughts on that. So going back to the baseline characteristics, I've just added to that earlier table, I've shown you the obesity proportion already, 20%. And then here is the baseline rilpivirine and cabotegravir resistance. This was surprising to us. 13% with rilpivirine resistance, 16% with cabotegravir resistance. Of course, this is archived virus. It may be non-replication competent. We're trying to do some more work on this, or at least figure out what this might mean. But certainly, it's suggesting that a number of these participants in the trial had archived resistance. And then when we look at the combined factors, it's really, if you have 2 factors or more, that you're at risk. So if you just look at the towards the end of this table, 2 factors present, got subtype A1 and rilpivirine resistance in, let's just look at the overall column, 7%, rilpivirine rams and obesity 1%, subtype A1 obesity. But if you take out the A1, if you say, well, actually it's just A6 and A1's not at risk, we're really just left with this combination of rilpivirine rams and obesity. And actually in the long-acting group, there was no one who had that combination. So, you know, it may be that without the subtype risk, this is actually an okay intervention, and these other factors are not really hugely important. So what additional evidence do we need for cabotegravir or rilpivirine to roll out in public health approach? Clearly, participants with adherence challenges, so this was done in a trial, people who are fully suppressed, so on, well selected, and potentially these you know, that's a good group to test this. But of course, with people with adherence challenges, what we know from the recent trial is that these are potentially a group that might do better on long-acting than standard of care. And there is a trial in sub-Saharan Africa called the Impala trial, which is testing cabotegravir rilpivirine in a group with adherence challenges, which is now fully enrolled as of last week, trial led by Fiona Cresswell. And I think that's going to provide very important complementary data to CARES. And then the hepatitis B issue is a hot potato, really. So all trials up until now have excluded participants with surface antigen positive and evidence of exposure with anti-hepatitis B core. But we know that the risk of reactivation with hepatitis B core positive is quite low. And if you've got anti-hepatitis B surface antigen positive at the same time, which most of the people in sub-Saharan Africa will have because they've had prior exposure, then that risk drops down below 1%. So, you know, thinking about the risk benefit of that and the potentially a monitoring algorithm is the way forward. And I'm pleased to say again that Impala learned from the experience with CARES and is including people with core antibody positive if they're also surface antibody positive. And then, of course, costs is a key consideration. And we really think about, what are the program benefits of doing this, and we can discuss a bit more. I don't know what I think it was supposed to be 20 minutes, so I think I may be over. But let me I'll just skip through these just to mention the other thing that I think is moving forward very actively is long-acting injectable cabotegravir for PrEP. That's now a WHO recommendation, and there was some very compelling data presented at CROI by Moses Kamya from Uganda, randomizing people to standard oral PrEP or post-exposure prophylaxis for TLD available at local Ministry of Health clinics versus the same intervention, but with the option of cabotegravir long-acting, again available from Ministry of Health clinics. And it was really dramatically increased the biomedical time when they were covered with one or more prevention measures, and even more so at times of HIV risk. And that translated into a clear benefit in terms of reduced HIV infections. So really compelling data and movement in this direction. So already it started in Zimbabwe, Zambia, Malawi, and PEPFAR is planning to roll this out, which no doubt they'll do very quickly and successfully. So that's very exciting. I might skip the lenacapavir because that's really for the future. So cabotegravir, rilpivirine long-acting. The CARES data supports there's high efficacy, a good safety profile, high participant satisfaction. A1 doesn't appear to be a risk, and obesity and rilpivirine on their own don't seem to have a clear association with outcomes, but the week 96 data will be important. Still a lot of things to consider, particularly hepatitis B, the logistics to support adherence. And then, as I say, cabotegravir happening, lenacapavir on the horizon, potentially of interest. Thanks very much again for the invitation.
Well, thanks so much, Nick, for those really important data and for taking us through a deep dive into the CARES study. I see that we're getting lots of questions coming in through the Q&A widget, so please continue to do that so that we can accumulate them and then give them to our panelists as we get to the panel discussion at the end. So it's now a real pleasure to introduce our next speaker, Roger Paredes. Roger is the head of infectious diseases department at the Hospital Germans Trias Pujol and principal investigator of the microbial genomics group at the IRSI-CAIXA AIDS Research Institute in Badalona, Catalonia in Spain. He received his MD and PhD degrees in medicine and surgery from the Autonomous University in Barcelona, and actually spent several years in my laboratory as doing his dissertation work, and remains to this day one of the most productive postdoctoral fellows to come through the lab. His team at IRSI-CAIXA has demonstrated the clinical utility of deep sequencing of HIV in both high and low income countries, and is now leading pioneering research into the role of the gut microbiome and its role in pathogenesis of HIV, as well as chronic inflammation more generally. During COVID, Dr. Paredes has been the Spanish national coordinator of really critical NIH and NIAID funded trials, including ACTS 1 and 2 and the Active 3 TICO platform, which helped define the standard of care for hospitalized patients as well as outpatients with COVID-19. Dr. Paredes is a member of the COVID-19 treatment guidelines panel of the Spanish Infectious Disease Society and the Catalan Institute of Health, and a member of the WHO Global Clinical Platform for COVID-19 Clinical Advisory Group, and serves in the WHO's HIV drug resistance strategy steering committee, the HIV treatment guidelines group, and the European Laboratory Initiative. Today, Dr. Paredes is going to speak on alternative mechanisms of INSTI-resistance. Roger, always a pleasure to see you.
So thank you very much, Dan. It was also a pleasure to spend these 3 years in your lab, and they taught us a lot. And I'm happy to be back and also to present also alongside these fantastic group of people. So I'll try. So my talk will talk about targets and off-target INSTI-resistance mechanisms, and I'm using this analogy that I already presented in South Africa in the previous conference. When we think about our targets for INSTIs, for example, these are the regions in the HIV genome that we usually sequence. So we are interested in the protease, in the RT, and in this case, in the integrase region, and also sometimes we sequence the V3 loop, for example, for the envelope. And this is what we usually do. However, when focusing on INSTI, in INSTIs, what happens is that sometimes we have patients developing virological failure to dolutegravir, for example, to TLD. And when we do a standard sequencing, we don't see resistance mutations. And then clinicians always think, and it's very legitimate to think, if there's something happening outside those targets. And to what extent those targets, what is happening in those targets could be affecting the clinical outcomes. And that's true. There are a number of usually off targets that where resistance or evolves also, and some mutations might accumulate and lead to some modulation of the resistant phenotype. And there are many off targets that we usually do not sequence, and here they are summarized. So, for example, there's the GAG, but this is important for lenacapavir or for the protease cleavage sites, the RT connection domain or the RNase H, which modulate resistance to some extent to NRTIs and NRTIs. But what is of interest for this talk is also other off targets, so targets outside the integrase, like the GP120 or the 3' polypurine track, where we know that there are some mutations might accumulate. The virus might evolve, incorporating different mutations at those sites. And that could be affecting the resistance, could be modulating the resistance to dolutegravir. So in the recent, a lot of research has been developed. But to make a long story short, this research is highly interesting in terms of the virology, and it also teaches, it is really amazing research. But you will see at the end how it is not necessarily absolutely impactful at this point in HIV in our patients and what we are seeing nowadays overall. So the first study that was presented, there was not a lot of data presented at CROI, but I would say that the key data presented were from Eriko Fried's lab by Yuta Hikishi. He presented an abstract at this year's CROI, and also there was another one in last year's, in the previous CROI. Essentially, but this has been recently published, and I have used the publication because the publication contains more details, and it's better, I think, for me to explain what they are observing, which is extremely interesting. So this is the title of their paper. And I will go I'll try to go stepwise to make sure that I'm able to explain what they are seeing. Here there's a first experiment. What they do, they culture a virus. In this case is a PNL43, which is an X4 virus from the lab. And they put it onto serial passages, and they keep passaging the virus with increasing concentrations of dolutegravir. And after many weeks, this is what they see. First, this virus, after approximately 20 weeks of passaging, it starts accumulating mutations in the envelope, which are shown in blue. Then, slowly, when they continue to passage this virus in the presence of dolutegravir, this same virus starts accumulating mutations, additional mutations in the envelope, some mutations in the nucleocapsid, a couple of mutations in the nucleocapsid that you will see. These are the zinc finger domains. And eventually, this virus develops two first mutations in the integrase, which actually do not confer phenotypic INSTI-resistance. But when they continue passaging that virus eventually develops a 263K mutation. But see, this is almost after 1 year of continuously passing these virus. When then essentially repeat this experiment, but looking at using a different, another virus. In this case, is an R5 virus, which is the CH185 virus, and they observe similar findings. So there is constant, after serial passages in the presence of increasing concentrations of dolutegravir, the virus starts to accumulate mutations in the envelope, then some mutations in the nucleocapsid, and finally some mutations, a few mutations in on the integrase. Because it is known that these envelope mutation, also D541, facilitates the evolution of this virus and confers a certain degree of also resistance to the virus. They passage the same virus also with the T151 mutation, and in this case, in the presence of dolutegravir, they see again accumulation of mutations in envelope, and finally the accumulation of the R32G in the nucleocapsid. And finally, they repeat the same experiment, but instead of using dolutegravir, they use increasing concentrations of islatravir. And what they see is that there's again accumulation of mutations in the envelope, although there's a relatively fast emergence of M184I, which is a mutation that's well known to confer low-level resistance to islatravir. So they want to know then more about what is that virus doing? What is the effect of those mutations, that accumulation of multiple envelope mutations that could eventually lead to high-level dolutegravir resistance? So the way they do that is that they create a clone, which is called 7X envelope, that contains this number of mutations that you can see here, mutations in GP120, mutations in GP41, but no mutations on the integrase gene. And then they measure the RT activity of a wild type shown in black circles, but also a mutant with an envelope mutant with 541, and also the 7X end. And what they see here is that in the presence of increasing concentration of the dolutegravir, on the left-hand side, you see dolutegravir 0 nanomolar, then 3 nanomolar, 100 nanomolar, and 1000 nanomolar. These virus that contains 7 envelope mutations is able to replicate in the presence of increasing concentrations of dolutegravir, whereas all other viruses are suppressed. So providing a direct evidence that this virus is resistant to the integrase inhibitors, in this case to dolutegravir. When they take all these areas in the curve and measure the fold resistance compared with wild type, what they observe is that this 7X and virus actually has more than 2,000-fold increased resistance to dolutegravir than compared to wild type. And then what they do additional experiments, and what they see is very interesting, because this virus that contains this amount of mutations on the envelope actually shows a reduced cell-free infectivity, and also a reduced relative fusogenicity. Okay, so the virus is accumulating mutations on the envelope. These mutations, in the absence of traditional canonical mutations in the integrase gene, this number of mutations accumulated in the envelope provide phenotypic resistance to dolutegravir in the context of serial passage assays with multiple cycles. But when they use also a single cycle assay, they don't see actually a difference. As I said before, that was an X4 virus, so they want to do the same thing using an R5 virus, which is these clones H414 and H316. And long story short, both of these clones are able to to replicate in the presence of high concentrations of dolutegravir. When they repeat the similar experiments as before, they see that these clones, again, that contain mutations in the GP120 and GP41, show a very high fold change in resistance relative to wild type, more than 4,000. And again, clear reduction in cell-free infectivity and reduction in relative fusogenicity of these viruses with a lot of mutations. Then what they do, they test, in this case, the 7X envelope virus, and they evaluate the fold resistance to other drugs. And what they observe is that these virus contains, well, it has high-level resistance to dolutegravir, but also to rilpivirine and cabotegravir, and also is associated with a modest fold change, resistance against FTC, efavirenz, rilpivirine, and even slightly to islatravir, darunavir, nelfinavir, and to a lower extent to lenacapavir T20, and no resistance to DMS 806 and to the alanine compound, which is also another integrase inhibitor. So their hypothesis is that this virus is, on one hand, is conferring increased resistance to a lot of mutations, increased resistance to dolutegravir, low cell-free infectivity and low fusogenicity. So their hypothesis is that these mutations that are being incorporated in the envelope are actually favoring an increased cell-to-cell transmission. And in order to prove this, they spinoculate sub-T1 cells, and they set up two types of experiments. One is putting the infected cells in a transwell next to target cells, but not together. So they are next to each other, but they are not, but they are physically separated. And on the other side, in the second experiment, they just put them together, and then they evaluate the cell-to-cell transmission. And what they observed is seen here on the left-hand side, left upper graph, is that when you put these cells together in a co-culture, actually there is an increase in infectivity when they measure P24. However, when they use the transwell system, such increase does not exist. And also the same thing for the clones, the R5 clones. Then on these experiments, D&E, what they use is they measure P24 positive cells in the presence of increasing concentrations of dolutegravir and rilpivirine. And what they can see is that whereas increasing concentrations of rilpivirine are able to eventually suppress all the three viruses, increasing concentrations of dolutegravir decrease the relative amounts of P24, but do not completely suppress the P24 for viruses with envelope mutations, particularly for the 7X envelope mutation. Then they do another experiment confirming that P24 signals are actually coming from the novel viral gene expression, as you can see here. This paper is a complex one, and I will just summarize why they do an additional testing to try to see what is the mechanism of these mutations. What are these envelope mutations doing to the envelope structure? And what they observed in the summary is that the ENV mutations alter the conformational landscape and stability of the ENV trimers. ENV variants exhibit higher CD4 binding and are resistant to choline activation relative to wild type. Increased populations of intermediate or high threat intensity in the unligated state relative to wild type. And also, these 7X ENV is relatively insensitive to conformational remodeling after interaction with CD4. So in summary, the accumulation of ENV mutations reduces CD4-induced conformational rearrangement of the ENV trimer, and this is necessary for a viral entry in cell-free infections. But it also stabilizes GP120 and GP41 interaction. In additional experiments, they then create site-directed mutants in different positions shown here. And what they observe is that a number of different mutants in the zinc fingers of the nucleocapsid are actually able to induce a certain level of resistance to dolutegravir in this case, although the mechanism is not fully explored here. And finally, their hypothesis is that, well, they think it's happening is that this virus, which is, being put under pressure by dolutegravir during a long time and during serial passages and very high concentrations of dolutegravir, this virus evolves, accumulates mutations. These mutations change the way these envelopes interact with cells, reduce cell-free interaction, but they promote cell-to-cell interaction of the cells. And what their hypothesis is that INSTI-mediated inhibition of HIV infectivity could be overcome by high MOIs, by high concentrations of viruses. So in this experiment here, first they calculate the high MOIs, and here what they see is that they measured virus production, the presence of increasing amounts of virus input. And what here is in the presence of dolutegravir, and on the left-hand side is in a wild-type HIV to start, and on the right-hand side also a catalytically inactive mutant. So they can see that when you essentially throw into the experiment a lot of viruses, these high amounts of viruses are able to overcome the effects of dolutegravir, whereas this does not happen when there's a catalytically inactive mutant. Then they go to evaluate different INSTIs. So the same experiment shown here is shown for rilpivirine and Cabotegravir, so essentially the same findings. However, when you can compare it with NNRTIs, NRTIs, and NRTTIs or PIs, this does not happen. These other drugs are less sensitive to high MOIs. So in conclusion, INSTIs are more readily overwhelmed by MOIs than other classes of antiretroviral drugs. And they finish there in the supplementary material. I actually use the same graph, summarizing what happens in the left-hand side when there's low MOI infection, when there's low amounts of viruses in these in vitro systems. So essentially the antiretrovirals are protecting these castle, which are the target cells, from the virus from getting in. However, when in a situation where there's a high MOI infection, when there's a lot of viruses, if they throw a lot of viruses, these viruses, in the case of integrase inhibitors, and particularly dolutegravir, they are able to overcome the effect of antivirals and then get into the cell and let's say create problems. So I hope I was able to explain these correctly and clearly enough. So this is a mechanism that is the study is very elegant. It's extremely elegant. This is something that's clear that happens, of course, and it's very well done. However, which are the public health implications of what they found? The public health implications are very uncertain. First, because there is no systematic evaluation of such mutations among dolutegravir failures. The prevalence or incidence of these mutations in real life in humans is really unknown. You need to keep in mind that they required more than 1 year to select those mutations in vitro under a very specific system. Also, there are a number of technical constraints. So envelope sequencing by itself is technically challenging. There are no geno-to-pheno associations. There could be multiple and diverse envelope mutational patterns that could lead to similar profiles or very different. Also, the phenotypic analyses are needed to evaluate this. So this is not detectable in single cycle phenotypic assays. Multiple infectious cycles are needed, so even the single cycle phenotypic assays might not be enough. And of course, the role of cell-to-cell transfer and multi-copy infection in people living with HIV remain a matter of debate, although it is generally accepted that this is something that occurs. I just took 2 sentences from the same paper saying that, well, first, we consider it unlikely that in vivo envelope mutations will confer the multi-log INSTI-resistance observed here, as highly efficient cell-cell transfer would likely lead to virus-induced cytopathic effect and detection of infected cells by the host immune system. However, even in the absence of multi-copy infection, mutations that increase the MOI would likely contribute to the acquisition of drug resistance, particularly in the context of INSTI-containing regimens. In other words, it is very uncertain that this is something that's happening very often or often enough to become a public health problem. However, this is an additional mechanism that could lead to modulation of INSTI-resistance and could facilitate INSTI-resistance in some patients, although most patients in whom we don't detect INSTI mutations are actually can be resuppressed, after adherence intervention. So we should not directly interpret that these patients actually have these other mutations. Another interesting mechanism in terms of, in vitro and for resistance studies is our mutations in the 3-polypyrene track. We know of those mutations since a paper published in 2017 by Mallet and colleagues from France, where they essentially infected MT4 cells with HIV live virus. And they cultured these virus with and without dolutegravir and maintained beyond initial viral breakthrough until 118 days. And then evaluated the RNA from supernatant throughout the experiment. And they found no INSTI mutations, but huge or high fold change for dolutegravir, raltegravir, and elvitegravir. And what they described when they sequenced these polypyrene track, they found a number of mutations in the polypyrene tag that they call the 6G pattern. And on the right-hand side of this slide, you can see how these patients that contain these mutations, that could be different variations of those mutations, actually had reduced susceptibility to the different integrase inhibitors, but that was not affecting the AZT, for example. Another finding comes from the mono study, in which this was a monotherapy study, and there were a number of patients who developed early virological failure. And Wisting and colleagues, they found one patient that had no resistance mutations on the integrase. They sequenced the polypyrene track and found also the presence of mutations in the polypyrene track during dolutegravir monotherapy. However, additional studies by the group by Nick Paton, they essentially found that either these viruses were these viruses by Mallet, a French group here in a square, had very low fitness replicative capacity. It was really very unable to grow. And the other one from the Dutch group from Whiting was associated with a small fold change against dolutegravir. There has only been an additional paper by the Decker and colleagues published recently in Journal of Virology, looking at the mechanism by which such mutations in the polypyrene track might be associated with resistance. And here on the left-hand side, you can see a normal, let's say a regular reverse transcription process, which is a multi-step process that includes several strand transfers and jumping from different areas. And here, the first part is the same, but what you see here in a circle on the right-hand side is the lack of these polypyrene track that cannot be used as a template to create a double-stranded DNA. And therefore, this leads the virus to form 1 LTR circles. And what they have observed is that these viruses can actually, 1 LTR circles can be an origin of continuous replication without integration. This is interesting in terms of mechanistically. So in summary, these are two sets of data which are extremely interesting from the virological perspective. However, the conclusions in terms of public health, which is, I think it's what is relevant to most of us here, is that there is no current need for systematic sequencing of off-targets for clinical or public health surveillance. On the first group is because the mutations in envelope or nucleocapsid, they appear after prolonged exposure in vitro. The mutational profile is hard to predict at the population level, and the clinical translation and the public health translation of such mutations is still very unclear. So we should not be really worried at the public health level. Regarding the polypyrene track mutations, again, these are extremely rare events in vivo that either lead to, so far, to low replication capacity or minor impact on susceptibility. And what we have learned is that resistance can happen through integration-independent replication via one LTR circuits. Let me finish by thanking José María Llibre, who shared some of the slides with me and helped me set these, structure this presentation, and also a number of groups with whom we work, and also all of you and that. Thanks.
Thanks so much, Roger, for those important insights. And it's certainly an area that we're going to want to be watching carefully, but it's reassuring to know that at the present time, we don't have to really be terribly concerned. Next, we're going to have a discussion between Professor Catherine Orrell and Professor Lloyd Mulenga on the impact of resistance on treatment strategies. Catherine Orrell is an HIV clinician and clinical pharmacologist who's been the principal investigator on more than 20 trials since 2004. She conducts research at the University of Cape Town, as well as in community settings and primary healthcare clinic environments, and leads a clinical research site in Guguletu. The site is currently conducting trials of novel methods to monitor and improve ART adherence and explore the impact of resistance. Professor Mulenga is an internal medicine and infectious disease physician currently serving as director of infectious diseases for the Ministry of Health and the Infectious Disease Division of the University of Zambia, and its teaching hospital, the University Teaching Hospital in Zambia. He serves on the United States NIH Data Safety Monitoring Board for NIAID-supported trials, as well as on the WHO Guidelines Development Group, the WHO HIV Drug Resistance Network, and the WHO Tropical Diseases Research Joint Coordinating Board, as well as the Zambia Tropical Disease Research Center. So let me turn this over to you to begin this discussion, and then as we move along, perhaps I'll pitch some questions, and then we can transition to the more general discussion.
Great, thank you. Lloyd and I haven't practiced this, so so I thought maybe there's things to talk about dolutegravir in programs, there's things to talk about cabotegravir in programs. So maybe I can start talking to dolutegravir a bit in South Africa, and then hand to Lloyd, and we can come back and talk a bit about cabotegravir, or the lack perhaps of cabotegravir in our settings. So for me, I mean, I spent a lot of time working on adherence. So it was always fairly clear to me after 20 years of ARV experience that we were going to break dolutegravir as well, somewhere along the way. And I think our job when we're rolling this out in programs is to try and minimize this for as long as we can and as carefully as we can. And I think that might boil down in my setting to more surveillance and looking at when we do our genotypings. And because I know we know that there is people who are on TLD2 on second line have more risk of resistance than people on TLD1. However, most of the people that I see don't know if they're on TLD1 or 2. We perhaps didn't have a viral load at switch. We have little ARV history. People switch from one clinic to another. So we have a mass of people who are taking TLD, and it's actually really hard to determine if it's their first line regimen or if it's their second line regimen. And therefore hard to sort of see how important is this raised viral load? Is this a first, raised viral load in someone on second line, or is this the first raised viral load in someone on first line that you might manage quite differently? So I think we need to be careful about every viral load. And certainly in the Western Cape, I know we're looking at the moment in our South African national program, we're only allowed to do genotypings in people who've been on dolutegravir for at least 2 years, however many times they've had viral loads that have been raised along the way. And we're looking at bringing that in a lot more tighter, so that when the first viral load is raised, you try and we would like to have an objective measure of adherence. So not just speaking to people about how they do their adherence, but perhaps checking their pharmacy refill, that they have actually had drug on board, making sure, or even Monica Gandhi's amazing urine tenofovir rapid assay, just to be sure, like you could check they're taking medication or they're not. That might change your thoughts about whether you head for a resistance test or whether you head for adherence improvement. How you improve adherence remains up for debate. In my mind, it's coming more towards making structural access to drugs better, making it easier for people to access medication, not have to spend the whole days in the clinic, rather than counseling people to take their tablets better. It's more towards differentiated service delivery, and making things easier for people than trying to sort of change behavior potentially. Yeah, so so I feel like we really need to be able to have an objective measure of adherence, have something pragmatic to do about improving their adherence, and then repeat the viral load and do resistance testing slightly earlier than we do at the moment in our programs. Handing to Lloyd.
Thank you, Catherine, and I surely I agree with you that we need to emphasize more on adherence, and also how do we monitor failure and resistance, especially that, as you have indicated, many of our people do not know whether TLD1 or TLD2. And we are now having a number of people who are recycling these molecules. When it comes to the issues around Cabotegravir-resistance, I think it's reassuring to have the CARES trial. Thanks, Nick, for sharing that. But we also mentioned one important point on the prevention. We are rolling out Cable in many of our countries. And so far, like in Zambia, when we started in February of this year. So far, we have seen some individuals who have, we did not pick HIV, early acute HIV infection using the antibody test, and we subjected them to Cabotegravir. We don't know, again, the implications over time. Even if many of those people, the time that when they are coming for the second dose, we picked using the nucleic acid test that they had HIV infection, which we would not pick using antibodies testing. The viral loads were very, very low, except one who had a high viral load, and we put them on TLD. So again, following up all these people that are having either breakthrough infections on Cab PrEP, or those that are having really missed infection and were exposed to Cabotegravir, is very key, and trying to see which mutations they develop over time. Another important point, I think, regarding resistance is these who are, especially for those on Cabotegravir, when they are exposed to Cabotegravir, we are largely, many programs, I think, are just putting them on TLD. Would the response be similar, or should we put them on a PI like darunavir? So I think those insights will be important to learn from countries. Rilpivirine as well. We have had a high use of NNRTIs in our settings, and many of the people that may be transitioned to use of Cabotegravir with rilpivirine may have also been exposed to NNRTIs. So the issues of recycling, I think there was an issue of recycling NNRTIs in the charts, a question. I think some trials did show that even when you recycle NNRTIs, accumulation of resistance also is high. I think I remember one legacy trial in children. So I think monitoring for resistance, whether we go to the injectable ARVs or the oral ARVs in the era of the INSTI, is very, very crucial for programs across the sub-Saharan African region. Thank you.
You've both touched on a number of really important questions. Maybe I'll ask the other speakers to turn their cameras on and join us. But let me ask you both, can you elaborate a bit more on what you see as the role of CAB rilpivirine long-acting in your treatment programs going forward?
It would be lovely to be able to elaborate on that, but we haven't had access to the drugs to test these things out. So for me, as a sort of South African who got left behind with ARV access 20 years ago, I'm feeling very similar. So we're getting left behind again, like the drugs registered in the country, there's no sign of CAB and rilpivirine for treatment. We get stuck in like, we can't. So it's a roundabout answer to your question, but when we don't know the cost of what these drugs are going to be, it's hard to work out who you're going to give them to. Because if we can dose 20,000 people a year, we're going to have to think very hard about who are those 20,000 going to be. But if we can dose 1 million people a year, then we can offer it to all adolescents who struggle with adherence all the time. I mean, I'm doing a small study now called Affinity, looking at drug use in Guguletu in our community setting, looking at long-acting injectables and mixing all the cohorts, a straight switch cohort, a struggling adherence cohort, and a naive cohort. It's more quality, feasibility, and acceptability. But these kids are loving this drug, like adolescents who've been on drugs for 16 years, are not having to take pills anymore, and it's fabulous for them. So adolescents would be one cohort, pregnant and postpartum women in that sort of postpartum chaotic period, to take away one set of drugs they'd have to take. That would be fabulous to do that.
And there's a trial that's going to be looking just at that question, right?
Yes. Yeah. Well, I have a grant to do one, but I can't get drug for my grant.
Oh,
so there's like all sorts of those kinds of issues to do. We were looking at postpartum bridging with injectables, and there's an ACTG study doing something very similar as well. Yeah. And then so that group, and then thinking my pharmacology brain in elderly people where, they've got lots of multiple other drugs to take as we move into the era of other comorbidities with people aging, maybe not having tenofovir in the regimen will be good for people as they get older and have renal issues and things like that. So, but I feel that we can't move forward in exploring all these options because we haven't got access to the drug. We haven't had data in, can we give this to people who are viremic? Does everybody really have to be virally suppressed? I have 6 adolescents out of 260 that we screened for the cohort who simply didn't get injections because they couldn't take their tablets. There was no other reason that they didn't get injections. It's just that they really struggle to take tablets. And I'm dying to give those young people these injections because they're exactly who needs it. But I'm forced by protocol to not give it to the people that need it. It's ridiculous. So for me, there's more activism with a rilpivirine, because I think unless you're AVA and your country is very forward thinking, we don't actually have access to these medications. So we can't answer these questions yet.
Yeah, no, and that was a limitation in the Latitude trial as well, that the fact that you first had to get people suppressed to be able to then put them on injectables, and that there are now a couple of trials ongoing or just starting about direct to injection for viremics. Lloyd, you mentioned the use of long-acting cabotegravir for prevention. One of the questions that's come up is about the implications of breakthrough on long-acting cab in terms of resistance and the potential role of rilpivirine in such persons, but more generally the role of integrase inhibitors. And you referenced that briefly. I wonder if you wanted to say a little bit more about that, or if others on the panel wanted to chime in.
Thanks, Dan. Indeed, first of all, we have included Cabotegravir-resistance for treatment in our program. And as Catherine did highlight, we can have guidelines. I think the beauty about our country is I think we are flexible enough to incorporate certain molecules even before WHO approval. That's how we even started with the CABLE for prevention. And we have a budget line from government as well towards this, but access is another problem. Last year, we failed to get these molecules because of lack of access for the prevention. And in the similar way, I think we see access as a challenge as well, even when we go for treatment for rilpivirine. We are hopeful that possibly this year, end of this year, we should be able to access those. And again, the question which you raise is, how is it going to play? Right now, our emphasis is that those that have breakthrough infections, we are trying to run away from use of integrase inhibitors and use the darunavir, especially that we are not in a research setting. This is in real-time implementation. But it will be good also to see which challenges come in outside the trial setting with the use of these injectables. We are hopeful that possibly by the end of this year, the population which we've looked at, adolescents, those that are having problems with adherence as well, and they are, once they are suppressed, then we move them to this injectable ART. And we also have some subpopulations, those that are on third line, that we think that they may also benefit, especially those that had no exposure to rilpivirine and also DTG, or who have been on DTG, but just for simplification of regimens. So these are the populations that we are looking at. But again, with the prevention, use of carb as prevention, I think we have to be very careful in how do we monitor these populations for us to learn more on the optimal regimens for them. Thank you.
Great. No, thank you.
Yeah.
Go ahead, Nick. Yeah.
Yeah, just a couple of comments on that. So, I mean, I think it's great. Zambia is ahead of many places in this. I detect that there's going to be a huge pushback on cabotegravir-rilpivirine. We have to see what WHO does in the forthcoming set of guidelines. But you know, I think both from the hepatitis B perspective, from cost perspective, until those things are worked out, and thinking big scale, what can this do for programs? That's going to be a challenge. Many countries aren't as proactive as Zambia, and will look to WHO for guidance on that. And it's a question you can't get to scale unless there's something that really the big agencies buy into. So I think that's going to be the problem. And just one second comment, if I may, Dan, just about the resistance, the rebounds on cabotegravir-rilpivirine. I think we don't yet understand enough about resistance in that scenario. We should not make the same mistake as with the nucleosides and just assume that somebody having a mutation is going to be a bad outcome. So I think it's critical in programs as well as in the trials to collect information. You get somebody with rebound, make sure you do a resistance test, document that. So we've got emerging data, and if it's obviously if you've got 118R or something, that person will not resuppress again. But if it's a 263K or just an isolated mutation, what is to be lost with giving dolutegravir and 2 nucleosides a go, and monitoring that carefully. If they resuppress, then great. If not, then darunavir and tenofovir 3TC, irrespective of previous failure, would be a very solid choice. Thanks.
Completely agree. There's been a lot of discussion in the Q&A chat about TLD1 versus TLD2, the duration of dolutegravir failure before resistance. So first of all, the question, I think this is for Gert, is should the transition to dolutegravir be limited to people who have no prior experience or history of treatment failure? Is that, or maybe you can clarify that, because that was a question that somebody raised.
I think the main message is that the TLD2, so treatment experienced patients, and I think it's clear from many studies with NRTI resistance, still have very good clinical outcomes on those regimens. I think the main public health message, so the public health message from public health perspective, I don't think it's justified to not do this because it simplifies treatment. There's good tolerance. But I think the one public health lesson is when they have virologic failure, we should not regard them as low risk for drug resistance if they have continued high viral loads. So it's not that single viremia that resuppresses, but the patients with ongoing viremia. And the other public health message is to move the first viral load, as Catherine said, to earlier. But another question that relates to this that I've seen in the Q&A is, what predicts resistance? Which are these patients? How many patients are patients who have TB and are on rifampicin? So that kind of data. And then what is the adherence pattern? We had the good data from Banksberg's group, who showed that with NNRTIs, it's those breaks in therapy, and with PIs, it's the overall pattern of adherence. But I haven't seen anything similar to DTG, especially because I think the rates of resistance were so low. So I think Nick might sit on some data. I think Catherine,
I'm getting some.
We might be generating some. I mean, your people are generating some, but I think this is really important to know what is predicting this. So that appeared from the Q&A. I don't know if any of you could also help to answer that, because I tried to answer, but my answer is I don't know.
Anybody want to take a stab?
Well, based, I mean, in Nadia, we've had a fair bit of criticism not writing up in full the resistance data. So we are working on that and, you know, planning to get some next generation, do a comprehensive look at it, and hopefully we can get that out in the next 3 to 4 months. It's long overdue. And I think that will certainly contribute because we've got evolution of resistance over time and 9 people develop, you know, but I think the nucleoside backbone seemed to be relevant there. So a zidovudine-based backbone, which of course few people will be using now, was responsible for most of those resistance mutations. So the tenofovir 3TC dolutegravir just only went as far as a 263K. So, you know, that was encouraging. So I think the potency of the background drugs, we couldn't see a clear pattern with adherence accounting for that. But as you said in your talk, Gert, the second, you know, first line failure. So coming in with potentially compromised nucleosides, we saw tenofovir did better than zidovudine, even though you wouldn't expect it in resistance. But it's not to say it's perfect. You know, I think these drugs are still compromised.
Roger, do we know if any of these envelope mutations that the Fried group identified are seen arising naturally or selected by other drugs, or is this only seen in the setting of selective pressure from integrase inhibitors?
No, I think some of those were actually also selected by islatravir in the same study actually. So the envelope is a highly plastic protein and essentially a molecule that adapts and finds combinations as long as the 3-dimensional structure is good enough. Possibly that's too simplistic, but the idea is that the envelope is finding, well, the virus is having a challenge. It tries to replicate, and then in the presence of dolutegravir, these mutations can happen, but they can happen also in other viruses. But we shouldn't consider them as mutations that are canonical and can be put in, let's say, in the IAS USA list, for example.
And if you passage virus in the absence of drug, I mean, could this just be further adaptation to replication in culture?
Some of them as well, although it seems that in the presence of these challenges, they happen faster. Although people have to know that these have been passages for more than 1 year. So this is really long experiments.
Because people, at least up until now, who've gone on to long-acting cabotegravir have been suppressed, much of the information about pre-existing resistance comes from doing proviral DNA resistance testing, which is a controversial area. I wonder if people want to comment on whether folks being considered for long-acting injectables should have resistance testing done first. It should relying on proviral DNA. That's certainly a controversial question for us here in the United States, where we have some access to that.
So I might start because I'm known to be controversial in this sense. I don't like proviral DNA for guiding resistance, so therapeutic outcomes, for therapeutic decisions. That's because it has never been validated as such. I think it can provide important information, but there's a lot of things that make sense that then don't happen, like Dr. Paton's studies have shown us. And I would be very careful in using proviral DNA without linking that into really solid outcome studies. That's my personal positioning, and I'm not shying away. Yeah.
I would agree
support
with that.
Yeah.
I think we might have consensus here about something.
Yeah, I can't see it happening in our programmatic setting for one thing. We like to learn from what you do.
And that's also, yes, it's also because if you find those mutations, there could be in dead viruses and then mean nothing. Just to explain to the audience, just to make sure. And also maybe you don't find them and they're there and they're also relevant. So anything that we decide clinically has to be really linked to good studies, and be very careful about that. Yeah.
Yeah. I think we need a new, sorry.
No, you go ahead.
I think we don't yet know if a new generation of assays that look at long intact proviruses might be more predictive. But until we have methods that are scalable, and we validated that, we really can't say what it means. I agree.
Can I just make one point for the defense? I'm actually in agreement with you all, especially from a pragmatic point of view. But you know, the analyses of FLAIR and ATLAS have shown a significant relationship between rilpivirine archived mutation and outcome. So there is some data there that show that association against confirmed virological failure. So you know, it may tell you something, but you've got to sort out from all the noise of the replication incompetent virus and so on. So it, you know, just may be a very too complicated to interpret and very low sensitivity and specificity, or what would be positive predictive value, I guess.
Yeah, it's interesting in those analyses, even though clearly there was a very high risk, still the majority of people with pre-existing mutations did suppress. And so it's another one of these issues of relative versus absolute risk. And yeah,
it depends also on the options you have, and of course how much you gain. So certainly this is the low-hanging fruit. If you have rilpivirine-resistance mutations and you have other options or better options, then that's fine. I think it makes sense to try something else. But then if you have to plan your programs, and depending on the specific context, you would be taking a lot of people out. And I just need to see more data on the actual outcomes to understand what is the size of that effect.
There was a question that came up that I think might be important to address, just to help put everybody at ease, and that was about whether viral subtype affects the response to treatment in general. You know, from my read of the literature, the answer is no, that we've fortunately seen that there's universally similar responses to whether it's a non-nucleoside or protease inhibitor-based regimen or an integrase inhibitor-based regimen. So I don't know if others want to comment on that. So,
no,
yeah,
I agree.
That's why it was so surprising that this initial signal emerged with the 6 viruses in ATLAS and in FLAIR. Let me see, what other questions should we bring up here? We've talked a bit about the major reasons for development of drug resistance, or at least the emergence of resistance mutations. I think we, you know, in general, I think we know that there are pharmacologic factors, especially drug-drug interactions, with rifampicin being the most important one, and then intermittent adherence. I think we had some discussion already about the differences in adherence patterns, and the data, the older data, a lot of which rely on multi-tablet regimens, and now we're in the universal single-tablet regimen era, so things might be quite different. So I'm getting a message that we need a little bit of time to the organizers to help wrap up this meeting. So let me thank all of the speakers for your excellent presentations, and the panel for this terrific discussion, and as well as thanking our audience members for submitting such thoughtful questions. I hope we've had a chance to address a majority of them, even though there's not time to address them all. And let me turn this back now to Elliot and Ava.
So thank you, Dan, and thank you to the whole panel. This was really a wonderful, very insightful presentation, a series of presentations and discussion. And I want to emphasize that this is what the April 2025 meeting will look like. It's a bunch of people sitting around talking in front of other people and answering questions and discussing, you know, different personal experiences and personal opinions, because no one knows, when it comes to this subject, no one knows what the absolute truth is, and maybe never will. And so we can only do the best we can from a public health perspective. So again, I encourage you to seriously consider joining us in Cape Town in April 2025, but more importantly, we plan to have similar sessions like this over the course of the next year in run-up to the meeting. And also, I encourage you to strongly consider looking at what kind of clinical or other research that you're doing, and seeing if you want to form that into an abstract to present at the meeting. So with that, I think we will close, and we will keep you posted on, we're interested in feedback, and we'll keep you posted on the next session that hopefully will happen in the next 2 or 3 months, okay? Thank you very much, and thanks again to all the panelists. Ava, do you have anything else you want to say?
No, ditto. Many, many thanks. It was a great panel. And, definitely submit your research so we can continue these discussions.
Thanks. Night.
Thanks. Bye-bye.
Thank you. Bye-bye.
Thank you. Bye.